In the early 1980s, astronomers at the University of California proposed to build the biggest, most powerful telescope in the world, with a projected cost of some $75 million. When the Max Hoffman Foundation pledged $36 million, a grateful UC agreed to name the project for its generous benefactor. Unfortunately, this way of saying thank you complicated the business of raising the remaining money. Other potential donors were reluctant to put up funds for a telescope already named for someone else, so the project stalled. Eventually, a second, much wealthier California philanthropy, the W.M. Keck Foundation, stepped in with a pledge to underwrite the entire project. UC was happy to accept, Hoffman or no. The new Keck telescope on the summit of Mauna Kea in Hawaii would be fully operational by May 1993. Unprepared to play second fiddle to Keck, the Hoffman Foundation withdrew its pledge, and UC administrators sensed a $36 million opportunity. In particular, Robert Sensheimer, chancellor of UC Santa Cruz, realized that the Hoffman money could bankroll a major project that would put Santa Cruz on the map. Sensheimer, a biologist by training, was keen to see his field enter the major leagues of big-money sciences. Physicists had their pricey super-colliders, astronomers their $75 million telescopes and satellites. Why shouldn't biologists have their own high-profile big-money project? So he suggested that Santa Cruz build an institute dedicated to sequencing the human genome. In May 1985, a conference convened at Santa Cruz to discuss Sensheimer's idea. Overall, it was deemed too ambitious, and the participants agreed that the initial emphasis should instead be on exploring particular regions of the genome that were of medical importance. In the end, the discussion was moot, because the Hoffman money did not actually make its way into the University of California's coffers. However, the Santa Cruz meeting had sown the seed. The next step toward the Human Genome Project also came from deep in left field. The U.S. Department of Energy, DOE. Though its brief naturally concentrated on the nation's energy needs, the DOE did have at least one biological mandate: to assess the health risks of nuclear energy. In this connection, it had funded monitoring of long-term genetic damage in survivors of the atomic blasts at Nagasaki and Hiroshima and their descendants. What could be more useful in identifying mutations caused by radiation than a full reference sequence of the human genome? In the fall of 1985, the DOE's Charles DeLisi called a meeting to discuss his agency's genome initiative. The biological establishment was skeptical at best. Stanford geneticist David Botstein condemned the project as DOE's program for unemployed bomb makers, and James Weingarten, then head of the National Institute of Health, NIH, likened the idea to the National Bureau of Standards proposing to build the B-2 bomber. Not surprisingly, the NIH itself was eventually to become the most prominent member of the Human Genome Project coalition. Nevertheless, the DOE played a significant role throughout the project, and in the final reckoning would be responsible for some 11% of the sequencing. By 1986, the genome buzz was getting stronger. That June, I organized a special session to discuss the project during a major meeting on human genetics at Cold Spring Harbor Laboratory. Wally Gilbert, who had attended Sensheimer's meeting the year before in California, took the lead by making a daunting cost projection. 3 billion base pairs, $3 billion. This was big money science for sure. It was an inconceivable sum to imagine without public funding. And some at the meeting were naturally concerned that the megaproject, whose success was hardly assured, would inevitably suck funds away from other critical research. The Human Genome Project, it was feared, would become scientific research's ultimate money pit, and at the level of the individual scientific ego, there was, even in the best case, relatively little career bang for the buck. While the HGP promised technical challenges aplenty, it failed to offer much in the way of intellectual thrill or fame to those who actually met them. Even an important breakthrough would be dwarfed by the size of the undertaking as a whole. And who was going to dedicate his life to the endless tedium of sequencing? Stanford's David Botstein, in particular, demanded extreme caution. It means changing the structure of science in such a way as to indenture us all, especially the young people, to this enormous thing, like the space shuttle. Despite the less than overwhelming endorsement, the meeting at Cold Spring Harbor Laboratory convinced me that sequencing the human genome was destined soon to become an international scientific priority, and that when it did, the NIH should be a major player. I persuaded the James S. McDonnell Foundation to fund an in-depth study of the relevant issues under the aegis of the National Academy of Sciences, NAS. With Bruce Alberts of UC San Francisco chairing the committee, I felt assured that all ideas would be subject to the fiercest scrutiny. Not long before, Alberts had published an article warning that the rise of big science threatened to swamp traditional research's vast archipelago of innovative contributions from individual labs the world over. Without knowing for sure what our group would find, I took my place along with Wally Gilbert, Sidney Brenner, and David Botstein on the 15-member committee that, during 1987, would hammer out the details of a potential genome project. In those early days, Gilbert was the Human Genome Project's most forceful proponent. He rightly called it an incomparable tool for the investigation of every aspect of human function. But having discovered the allure of the heady biotech mix of science and business at Biogen, the company he had helped found, Gilbert saw in the genome an extraordinary new business opportunity. And so, after serving briefly, he ceded his spot on the committee to Washington University's Maynard Olson to avoid any possible conflict of interest. Molecular biology had already proved its potential as big business, and Gilbert saw no need to go begging at the public trough. He reasoned that a private company with its own enormous sequencing laboratory could do the job and then sell genome information to pharmaceutical manufacturers and other interested parties. In spring 1987, Gilbert announced his plan to form Genome Corporation. Deaf to the howls of complaint at the prospect of genome data coming under private ownership, thus possibly limiting its application for the general good, Gilbert set about trying to raise venture capital. Unfortunately, he was handicapped at the outset by his own less than golden track record